As the moderator of today's education, I'll be walking you through this virtual program. Before starting the program, I would like to introduce to the orator of today's topic, Ms. Monica Joshi. She is pursuing her PhD in Computer Sciences from SIMU. She did her M.Com in Pharmaceutics from Rajat Academy of Pharmacy. She has both academic and industry experiences. She has a total of 10 research publications, one book chapter, and one book. She is going to talk on the topic need of standardization of herbal medicine for improving Indian system of medicine, thereby enhancing global acceptance. Ma'am, please enlighten us with your. Ma'am, please.
Ma'am, I'm not able to share my screen. Please, host has disabled the attendee screen sharing. So kindly allow me to share the screen. Okay. Is my presentation visible, ma'am? Hello. Is my presentation visible?
Yes, ma'am. Yes, ma'am.
Okay, thank you, ma'am. Thank you. So for the today's faculty development program, I want to deliver my lecture on need of standardization of herbal medicine for improvising Indian system of medicine, thereby enhancing the global acceptance. Since the drug development process starts from a newly synthesized compound to a marketable compound, it takes around 10 to 15 years, and it also involves lots of money. Around 1 billion of money is involved during the drug development process. So a lots of money is involved, a lots of time is involved, but the success rate is very low. Most of the compounds get fail in the late phase of the clinical trials, and some are also disapproved after getting the marketing approval. So there is a need for the development of the compound that will take less time, and that will involve the less amount of money. The time tested least traditional medicine involves less cost and less time following the reverse pharmacology process. And if we are going towards the Indian system of medicine, which is mainly based on Ayurveda, that have its origination roots from the India only, but where we are lacking is in the standardization terms. Therefore, standardization is very important so that we can enhance the global acceptance of our traditional medicine, of our Indian system of medicine. So moving toward the next slide, the WHO has defined herbal medicine as the finished label medicinal product that contain active ingredients of the plant, aerial or underground part of the plant, or other plant materials which can be in the combination also. And as per World Health Organization, standardization of herbal medicine is the process of prescribing a set of standards or inherent characteristics, constant parameters, some definitive qualitative and quantitative values that carry an assurance of quality, efficacy, safety, and the results generated are also reproducible. So if our product is standardized, we can assure that it, it is effective, safe, and is of good quality. So standardization in this term is very important. The herbal drug is a broad term that includes Ayurvedic herbalism, also Chinese herbalism, and Western herbalism. Ayurvedic herbalism is derived from the Sanskrit word Ayurveda, that means the science of life, and this has its origination in India more than 4,000 years ago. While the Chinese herbalism This is an element of traditional related medicine which has its source of origin from China. Third is the Western herbalism, which is originated from Rome, Greece, and then multiplied to North Europe and South America. Here, I am more concerned towards the Ayurvedic herbalism as it is based on the Indian system of medicine. Now, when we standardize the herbal drugs, there are certain problems applicable to the herbal drugs that are not applicable with the synthetic drugs. For example, the herbal drugs, and this has rich history dating back over 5,000 years. That this traditional system of medicine is based on the belief that health and wellness depend on the delicate balance between the mind, body, and the spirit. And the current scenario of the herbal medicine in India is a cause of concern as, according to a recent study, over 60% of the herbal medicine that are sold, that are marketed in India, does not even meet the basic quality standards. This includes the contamination with heavy metals, toxins, inconsistencies in the concentration of the active ingredient. And if they are not properly regulated, they are not properly standardized, it will be very difficult to assure that these products are safe and these products are effective. So, to increase the global acceptance, one should standardize the prepared herbal formulation. And if we compare the global acceptance of our Indian system of medicine with the other traditional system of medicine, like the other traditional medicine systems, such as traditional Chinese medicine, it have gained global acceptance due to their standardization efforts only. For example, the World Health Organization has recognized the importance of traditional Chinese medicine and therefore has included it in its international classification of disease. And this recognition has helped to increase its global acceptance and promote its use in healthcare setting around the world. Therefore, if we will standardize our Indian system of medicine, if we will follow the standardization parameters properly, then we can also improve the global acceptance of the herbal medicine that are developed, that are formulated in India. Since there is a great demand for the plant derived products in the developed countries as well, and these products are increasingly being sold as a medicinal products in pharmaceutical industry, in nutraceuticals industry, and in cosmetics also. And when these traditional medicines were developed, the technology and the concept of standardization was quite different. At the time, when we moved to 4,000 years back, at the time, Veds, Hakims were used to prepare the medicine as per the requirement of the patient. And those were their quality that were evaluated by the Veds and the Rishis at that time. But since the commercialization has increased, these products has to be distributed to the longer distance, even the climatic changes have occurred, the soil have changed, the environmental conditions are now different. Therefore, these plants materials need to be standardized. These property of botanicals may also have undergone changes, and therefore the standardization is very important. And due to the commercialization, due to the increased demand, the supply of genuine material at current scenario has become a major challenge also. So in the current presentation, in the next upcoming slides, I will cover the various standardization parameters as per the WHO guidelines or the Ayush guidelines that comes under the Ministry of Health and Family Welfare. So as per these guidelines, the standardization involves three steps. Step is the standardization of the raw drug. Second, the standardization of the extract derived from that crude drug. Third involves the standardization of the formulation or the finished product that contains the extract. So these three steps should be followed for the standardization of the herbal preparations. The first is the standardization of the raw drug or the single plant material. So these are the parameters that one should include during the standardization. That includes botanical description, organoleptic description, authentication that one should conduct, foreign material presence, total ash determination, pesticide residue, microscopic characters, moisture content, extractive values, foaming index, particle size, shelf life, dose and manner of administration. And in botanical description, one should mention the Sanskrit synonyms, the regional name of that plant, the parts of that we are going to use to prepare the formulations, the botanical origin of the plant, its distribution, adulteration, substitution, the season of collection that also affect the quality of the plant material should also be mentioned. The site of collection, the stage of the plant at which we have collected, substitution or exhausted products and the foreign material presents. Further, the organoleptic second parameter, the organoleptic description is the most easy way to identify the plant that involves the color, odor, taste, and texture of the plant. For example, in case of Mentha piperata, one can smell out the presence of the menthol and can identify it easily. So these are the most easy organoleptic evaluation. Further, the identification of the plant should be carried out. Authentication should be done by the botanist, and the identification parameters for its microscopy studies, the color, its texture, through which we can identify the plant. Then the packaging and the labeling details, the Ayurvedic and the herbal formulation in which it has been used should be mentioned as per the Ayurvedic Pharmacopoeia of India. These are the some permissible limits for the pesticide residue since the plant material is derived from the soil from where the pesticides have also been transferred. So there are the permissible limits that has been given by the WHO. So it, this should be as per the WHO guidelines. Some of them I have included in the current slide. For example, Elacor, it should be under the 0.02 mg per kg, then bromopropylate 3 mg per kg, chlorpyrirox 0.2, DDT 1, endosulfan 3, hexachlorobenzene 0.1, malathion 1, and parathion 0.5. Further, second step involves the extracts, standardization of the extract that we have derived from the plant material. So the as standard operating procedure for the preparation of extract should be as per the Ayurvedic pharmacopoeia or as per the guidelines. That involves the raw material which we have collected should be grinded for the size reduction, and the solvent used for the extraction should be mentioned as per the guidelines. It can be water, alcohol, supercritical fluid, or a mixture of these. Further, the method of extraction must be followed as per the guideline. Either it may be maceration, decoction, or soxhlate extraction. Then the extract should be concentrated under the vacuum using the rotary evaporator, followed by drying on water bath or under vacuum. The quantity of the extract obtained is determined and the percent yield is calculated. Further, the extract obtained as per the SOP standardized operating procedure should cover the following standardization parameters like identification, physiochemical parameters, residual solvent if present, preservatives if we want to preserve the extract for longer duration, then type of preservatives we can use, assay for the constituents, test for heavy and the toxic metals, authentication, test for specific pathogens, pesticide residue, test for aflatoxins, shelf life, packaging and the labeling. So the SOP of the extract should be followed as per the Ayush guidelines, and its description, color, odor, taste and texture must be mentioned. The extract should be carried out, the identification test should be carried out that can involve the various phytochemical screenings like alkaloid test, glycosides test, or presence of tannins, presence of others groups, and TLC, HPTLC or HPLC must be conducted. And it has been mentioned in the guidelines that the HPLC or HPTLC must be conducted under against the three reference standards. Minimum three standards should be used to identify the constituent present in the extract. Further, loss on drying, total ash determination, acid insoluble ash, water soluble extractives, alcohol soluble extractive. If water soluble extractive and alcohol soluble extractives are less, it means that the plant is more adulterated. That should also be determined. Presence of heavy toxic metals should also be determined, and their limits are also given as per the WHO guidelines, as per the FDA. Then microbial contamination must also be evaluated, and there are the tests for the specific pathogen that are given in the Ayurvedic Pharmacopoeia of India, in the WHO guidelines that one should follow, like for the E. coli, Salmonella, S. aureus, or Pseudomonas aeruginosa, that should not be present. Aflatoxin, which is the fungal product, which is the fungal byproducts, should not be present in the plant material, and if it is present, it should be removed by the radiation techniques available. Now, third step involves the SOP of the finished product, the standard operating procedure for formulating the finished product. All standardized ingredients that are needs to be mixed should be as per the formulation, and these should be mixed under the hygienic conditions. These can involve the raw material plant extract, the bhasmas, and as per the Ayurvedic preparation like avleh, avleh is the paste-like preparation, then asav, which is the fermented syrup, churn, then quath, which is the decoction, then vati, which is known as tablet. So these are the most commonly used formulation that are commercially available right now in the market and that are mostly prepared. So their procedures were mentioned in the Pharmacopoeia of India, and that should be followed for the preparation of these formulations. Standardization of the formulation depends on the type of formulation that we are preparing. Like if it is vati, then its parameters will be different, and if it is quat, its parameters will be different. So the SOP of the formulation one should follow. Then particle size determination is important for the in case of churn. Disintegration time in case of tablet should not be more than vati. It should not be more than 15 minutes. Hardness, friability, then assay for the constituent must be conducted as per the guidelines. Test for heavy or any toxic metals if present. The procedures are given in the Ayurvedic Pharmacopoeia of India that one should follow. Microbial contamination, test for any specific pathogens, test for aflatoxin, these must be followed. Further, the lethal dose of that product should be determined. Optimum effective dose should be determined. The mode of administration, the shelf life of the formulation that has been prepared must be carried out as per the guidelines. Then packaging and labeling, the labeling part must mention all the formulation ingredients that we have used while preparing the formulation. Further, it also contains the formula used for preparing the formulation. The description, color, odor, and taste of the formulation must be mentioned. Identification test must be carried out. Then the physiochemical parameters that includes the friability, the hardness, the disintegration test, the uniformity of weight test, pH determination must be carried out as per the Ayush or WHO guidelines. Further, there is one SOPs for the anti-diabetic herbal doses form that I have mentioned in the current presentation. That like it involves the collection of the raw materials, then the cleaning of the raw materials for the removal of any dirt or the dust if present. Then the raw material is grinded for the size reduction. Then the purification is carried out, followed by drying of the extract through lyophilization process, and then these extracts are made free flowing. Then these extracts are filled in the capsule for the making the finished formulation, followed by packing of that capsule, labeling, and the storage condition, and then at last the finished product is prepared. So at last, in conclusion, I want to say conclude that standardizing herbal medicine in India is crucial for enhancing the global acceptance of Indian system of medicine. By ensuring safety, efficacy, and quality control, we can establish trust and credibility among consumer and the practitioners also.